Hello everybody. Today we are going to talk about one of my favorite topics, which is metabolism. It's also one of the most commonly heard words, but one of the most misunderstood. If you want more information or you just want to know where to look in the textbook to get additional examples, please refer to Chapter 6 in Biology in Focus. As with every unit, I will give you a general list of need-to-knows. These are always kind of your basic needs-to-knows. Make sure that you have specific examples and understand the details. So the first thing that we will talk about is metabolism. Metabolism is basically defined as the total sum of chemical reactions in the body of an organism. These can be used to manage materials insofar as breaking down and building things within the body, as well as affecting the energy resources in a cell, because anytime we break down or build something, we're either going to be using energy or releasing energy. In your need-to-knows, you're going to see that you need to really understand the differences between catabolic and anabolic reactions. Catabolic pathways, also known as catabolism, release energy, and they release energy by breaking down complex molecules into simpler compounds. One of my previous AP biology students once said that their favorite memory trick for remembering that catabolism has to do with breaking things down is that if a cat goes through a window, the window is going to get broken. So if that helps you to remember things, fantastic. A great example of a catabolic pathway is in the breakdown of carbohydrates. And so over here I typed some keywords for you. In your mouth, in your saliva, you will find an enzyme called amylase. And amylase is used to break down starch into smaller units called maltose and dextrose. These are further broken down in the small intestine. So we'll type that in, small intestine. And the reason why these are catabolic, again, is because you're breaking down a very large carbohydrate, which is starch, into smaller subunits. And energy is released when this occurs. Anabolic pathways, on the other hand, use energy. And the way that I remember anabolic is used to build molecules is because A is an ad and A is an anabolic. So a great example of an anabolic pathway would occur in conjunction with a catabolic pathway. So let's say that you have a big juicy steak for dinner one night. If that steak is broken down into smaller subunits, let's talk in particular about the proteins in that steak. So the proteins in that steak might be broken down into amino acids. So is that catabolic or anabolic? That's right, it's catabolic. And if you forgot that it was catabolic, just think of the cat going through the window and breaking it. So catabolic, break things down. So if we're breaking down the proteins in the steak into amino acids, that's catabolic. Anabolic pathways, however, will use energy, and they're going to take those amino acids, and those are linked together by a specific type of bond that we learned earlier in this unit. Hopefully you remember that that's a peptide bond, and those peptide bonds link together amino acids to form polypeptide chains, which will eventually form muscle protein. One of the other key things that you need to know are the different types of energy. So different types of energy when we're talking about biological systems are kinetic energy and potential energy. So kinetic energy, if you think about kinetic, that means motion or movement. Kinetic energy, which is abbreviated KE, is associated with motion. Now heat, which is basically composed of thermal energy, is a type of kinetic energy, and that's associated with random movement of molecules. And heat is a very common type of energy that's associated with living things, and we'll be looking at that in a little bit. Now, potential energy, abbreviated PE, is stored energy, and basically the most common type of stored energy that we're going to see in living things is chemical energy. So chemical energy is basically stored energy that's available for release in a chemical reaction. keep in mind that chemical energy can be converted from one form to another, but it can never be created. So make sure that you write down, never created. A common mistake in biology that students often make is that they say, well, photosynthesis creates energy. No, it doesn't. It converts solar energy into chemical energy, right? So another example here would be chemical energy to mechanical energy to electrical energy. But in all of these cases, we're not creating energy. So this is an example that's in your book that talks about how energy can change or be converted from potential energy to kinetic energy. So it has a diver on a platform, and some of you are involved in diving and in swimming. Many of us have, while we don't do sports, maybe have dove off of a platform in our pools. So basically, standing on the platform and not moving, that diver has a lot of potential energy, that's stored energy. And as soon as the diver dives off, that potential energy is converted into kinetic energy. Now, if the diver is moving into the water to the platform, there is still some kinetic energy going on. So there's a lot less potential energy there because it's still moving. The diver climbing up the ladder is still using kinetic energy, but there's absolutely some kinetic energy from that muscle movement that's being converted. Another really great example that you might want to think of is a rock just sitting on the top of a hill. It has all potential energy and no kinetic energy. When it's rolling down the hill, it has both potential and kinetic energy. As it's really, gone down the hill, it's slowly rolling down the hill, it's rolling on the flat part after going down the hill. It has kinetic energy because it's still moving, but it doesn't have potential energy because it doesn't have that potential energy for more energy to be released. And once it's totally stopped at the bottom of that hill on that flat surface, it's not moving at all. So it doesn't have any kinetic energy, but in this case, it's on a flat surface. It doesn't have kinetic energy at all. Now, if we push that rock back up the hill, the kinetic energy of being moved up that hill is also being stored, and then when it's resting on the top of the hill, it's just that potential energy. So it's something of a cycle. So any time that we talk about energy transformation, we are going to be talking about thermodynamics. And there are two laws of thermodynamics that you really need to know in this class and to understand so that you're not someone who breaks the laws of thermodynamics when you're trying to answer a question on the AP Biology exam. So in order to really understand energy and to understand thermodynamics, you need to understand the differences between closed systems and open systems. So a closed system would be a system that has no connection with its surroundings at all. And that's really different than a biological system. So an example that I put in the PowerPoint is that the liquid inside of a thermos, so let's say it's a hot liquid, it's basically closed. That heat, that kinetic energy is contained within the thermos. Now, some people might argue that eventually that liquid does get cold. So you could argue that there is some contact with the environment where that heat energy is being released. And in that, you would be correct. But just for this example, let's say that it's totally closed and that heat would never leave. An open system is where you're going to have energy and matter that's going to be transferred between the system and the surroundings. So if you opened up that thermos, that heat energy would absolutely be released. Most likely you're going to be taking some of that warm beverage out and drinking it. So there would be a transfer between the heat energy as well as the liquid inside. We are considered open systems. We take in energy from the outside, and we release energy. Also, we take in and release matter. This is true of all organisms. let's take a look at some of the different laws of thermodynamics. So the first law is that energy can be transferred and transformed, but it cannot be created or destroyed. And this is also called the conservation of energy. So when you're discussing something like photosynthesis, where photosynthetic organisms take in solar energy and convert it into chemical energy in the form of glucose, this is a transformation. This is not creating energy. You're creating starch or glucose, but you're not creating the energy. The energy within that molecule has been transformed. Do not be the student who breaks the first law of thermodynamics on their test. The next law of thermodynamics that you really need to understand is the second law of thermodynamics, which basically says that anytime you have energy transfer or transformation, it increases the disorder, also known as entropy, of the universe. So basically, every time that you have some type of energy transformation in your body, some of that energy is not being used and it's lost as heat. Okay? And remember that heat is a type of kinetic energy. So anytime when you do, for example, cellular respiration, basically what you're doing is you are using glucose and you're creating ATP, which is used in chemical reactions, and you're losing carbon dioxide and water. And anytime that happens, you're releasing heat, and you can feel heat when you touch your body. Now, you might be wondering when you look at this example, how is it that a living thing is so orderly? Because remember, order and organization is one of the characteristics of life. So how can we be so orderly if we're constantly losing energy as heat, we're constantly increasing entropy and disorder? Well, both of these things can happen because basically order within living systems is going to be produced by using energy that is created by photosynthesis. So for organisms like us, heterotrophs, that don't transform energy from the sun, we take in energy that's created by photosynthesis in the form of glucose. So we'll eat things and we'll take in this very ordered type of energy. So basically, biological systems are made to be orderly in one way by photosynthesis. It's one of the many reasons why photosynthesis is so important. So ultimately, when we're talking about energy, we want to be talking about free energy. Because what's lost as entropy isn't really going to help that system or help us to perform work. So we have free energy. And if you look here, this delta in front of the G means change. And G is referring to Gibbs free energy, which is just another way of referring to free energy. So the change in Gibbs free energy is equal to the change in the free energy. So one of the concepts that you really needed to understand in the things you need to know are exergonic versus endergonic reactions. So an exergonic reaction is going to be when energy is released, or the way that I remember this, it exits the system. So I would definitely go ahead and put exits into your notes here. An exergonic reaction is when energy exits the system, and so the change in the free energy is going to be less than zero. An endergonic reaction is going to be when energy is required, so energy has to enter the system, and the way that I remember endergonic is EN, is enter. So basically, in this case, we're going to be taking in free energy, so the change in free energy is going to be greater than zero. Another thing to notice here is exergonic reactions are spontaneous reactions because the energy is released, no energy is required, so they can occur just blam. Whereas endergonic reactions are going to require energy coming into the system. One of the things with these types of reactions is it's really important that you are able to recognize these types of reactions by looking at a graph. So in this case, the exergonic reaction is where energy is released. So would that be catabolic or anabolic? So let's take a look at this. In this type of reaction, we have our reactants, and you'll see on this graph that there's lots of free energy, and then that free energy is decreased. So what type of reaction might this be? In truth, it would be a catabolic reaction. Remember, catabolic reactions would be those reactions where something is broken down and energy is released. A great example of this would be cellular respiration, or at least the first part of cellular respiration, where we are taking a glucose molecule and we are breaking that down. And by breaking that molecule down, the energy is going to be released. And ultimately, that energy is going to be used to make another molecule called ATP. But that first part of cellular respiration, where glucose is being broken down, that is absolutely catabolic. So let's look at an endergonic reaction. So in an endergonic reaction, there isn't a lot of free energy. Energy has to come in to produce the products. It is not spontaneous, and we absolutely need some energy. So would this be an example of anabolism or catabolism? What do you think? If you picked anabolism or an anabolic reaction, you are absolutely correct. So anabolic reactions are going to be reactions where we're building something. So we would need energy to form those peptide bonds to make those proteins. So cells basically need to use energy in three main ways. Cells do mechanical work. They also do cellular transport and chemical work. A lot of examples are included in your book, but I'll give you an example of each that you can add to your notes. So an example of mechanical work might be your motor proteins. So motor proteins are going to be what allow for muscle contraction, also the beating of cilia. If you remember from your introductory biology course, cilia are those fine, small, little projectiles from some cells that work together in a coordinated beating motion to allow for cellular motion in most cases. In some cases, it allows for molecules to move through the throat. It traps foreign particles so that the molecules can move through and not harm your lungs. Cellular transport, these would be things like proteins and pumps found in the cell membrane. And then we've been talking about a lot of different types of chemical work, so endergonic and exergonic reactions. Let's put exergonic down here too. So basically, one thing to keep in mind, for cells to use a lot of the energy resources, they use energy coupling. So you use an exergonic process to drive an endergonic process. And we kind of talked about that in a little bit of our examples, but let's talk about it in more detail next. So we've already gone through examples of endergonic and exergonic reactions, and the next thing that you really need to know is the role of ATP in energy coupling. So let's take a look. ATP stands for adenosine triphosphate, and you absolutely need to know how to spell that, and you really need to understand the structure. So ATP stands for an adenine, and while you don't necessarily need to know the exact structure of adenine, you should understand how the adenine binds to ribose. And adenine and ribose come together to form adenosine. Okay, so adenine and ribose form adenosine. Where else have we seen ribose? Does anybody remember? If you said RNA, you're exactly right. So ribose is not only found in nucleic acids, but it's found in ATP. And so adenosine comes from adenine and ribose, and then it's called triphosphate because there are 3 phosphate groups. So this is the basic structure of ATP. Now let's look at how is ATP used as the main energy storage molecule in the cell. And really, that is a function of ATP. It is the main energy source in your cell. So basically, energy is stored between the bonds of these phosphate molecules here. Now, to release that energy, the bonds are going to be broken between these 5 phosphate groups. Now, remember, when we're breaking bonds, it's a hydrolytic reaction. So we're using what molecule? If you said water, excellent job. Water, hydro here refers to water, and so water molecule is broken. And when that happens, the water molecule, the parts of the water molecule will bind to the phosphates, and energy will be released. And the release of that energy basically comes from the chemical change to a state of lower free energy, not in the bonds themselves. And this is important. So the energy is stored in the chemistry of these bonds, but it's not the bonds themselves. When that energy is released, the chemistry of the bonds changes. So hopefully that makes sense. Let's take a look here. So ATP performs work in organisms by releasing a phosphate group. So that's what's referred to here as PI. That's a release of a phosphate group. And that's used to do endergonic work. So basically, that phosphate is going to come in and it's going to attach somewhere. And we'll talk about some specific examples of this a little bit later. When ATP is hydrolyzed, that is, when that water comes in, and helps to break apart that bond, it becomes adenosine diphosphate because now there are only 2 phosphates and di is a root meaning 2. So that energy basically from breaking down this phosphate, that's a catabolic reaction, is going to be used in an endergonic reaction where it's going to consume that energy and it's going to build things. So this is absolutely a cycle. The reason why it's a cycle is because ATP can then come back in, a phosphate can then come back in after it's been used, and we'll use cell respiration to make it into ATP again. So this is a cycle. ATP is absolutely reusable. ATP is broken down, a phosphate, and that energy is used. Then that phosphate comes back in. It's attached back to ADP where we make ATP again. So basically, I told you that that phosphate would then go to be used for cellular work. So here are some examples. That phosphate will come in, and in the case of mechanical work, you'll see the phosphate attaching to the motor protein and bringing that energy with it. So you'll see here now we have some motion. This process of that phosphate attaching is called phosphorylation. So phosphorylation occurs when ATP phosphorylates the motor protein, causing motion. In transport work, the same basic thing will happen. The phosphate will come in, it will phosphorylate that membrane protein, and it will allow movement, which causes for that solute to be transported. And lastly, when we're talking about chemical reactions, you will see that that phosphate will come in, it'll phosphorylate the reactants, it will provide the energy it needs for the reaction to take place, and then we have our products. In all 3 cases, you'll see that the phosphate group is released. That phosphate group will be rejoined to an ADP. That ADP will then undergo cellular respiration to form ATP. And again, that ATP can be broken down. Energy will be released with the phosphate, and that phosphate will join in different ways in different cells to perform different things. So other key things that I told you that you needed to know is how enzymes work and the basic cycle of an enzyme and how enzyme activity can be influenced by environmental factors. So we're going to take a look at this next. So the basic definition of an enzyme is that of a biological catalyst. But what does that mean? A catalyst is basically something that can change the rate of a reaction without itself being altered. So enzymes, while they help chemical reactions, do not change at all in the chemical reactions that they help. So what do they really do? Well, basically what they do is they act to speed up reactions, but they don't do that by providing energy, and that's a huge misunderstanding. Enzymes do not provide energy. Instead, what they do is they lower the activation energy required to start the chemical reaction. So the activation energy is the energy needed to start the reaction. And basically, what an enzyme is going to do is it's going to lower the amount that's needed. So let's take a look and see how they work. And this is a great example for you to know is sucrase is an enzyme that works on sucrose. And it basically helps the sucrose to separate and form into glucose and fructose, which are the monomers of this disaccharide. So let's take a look and see how this works. So the first thing that you should be able to do is to recognize any graph of a reaction with and without an enzyme. So if you look here, here's our free energy, or the Gibbs free energy. This black line here is what the reaction would look like without an enzyme. And you'll see here, there's a lot of energy that's required for that reaction to start. So this is the activation energy, abbreviated EA, without an enzyme. When the enzyme is put in place, you'll see the amount of energy that is required to start the reaction becomes much lower. So you'll see a huge change in here. Now, this is basically showing the course of the reaction with the enzyme, without the enzyme. One thing that you're going to notice here is that the change in the Gibbs free energy, or the change in free energy, isn't what's affected by the enzyme. The only thing that's affected is the activation energy. Please keep this in mind anytime you're talking about an essay, you're talking about an enzyme in an essay, or you're talking about the enzymes in your lab report. So let's look at a little bit more detail of the enzyme. So the first thing that you want to notice is that each enzyme is specific to a specific substrate. So this is kind of a little cartoony picture which shows you the chemical fit of the substrate to the enzyme, when in truth it's not like a jigsaw puzzle, it's an actual chemical fit. Now, the thing that the enzyme works on is called the substrate. The area on the enzyme, and this yellow thing is the enzyme, so I would definitely label that as the enzyme. The place where the enzyme binds is called the active site. So that's the region where the substrate binds. And when the substrate is in place onto the enzyme itself, this is called the enzyme substrate complex. So that's when those two things are grouped together. So let's look at another picture that's a little more dynamic. So in this case, we have substrates that enter the active site. So remember, the active site is this area here. So the substrates have entered the active site, and they're basically held in place by some weak interactions. And then you'll see that this active site is lowering the activation energy and therefore speeding up the reaction. The substrates at the active site are converted into the products, and then those products are released. A really good example of this would be lactose and lactase. So let's take a quick little look at this. I don't have a picture, but let's go back to this picture here. So lactose is basically a disaccharide, lactose, and it is composed of two simple sugars, galactose and glucose. So basically, if this molecule here, you could say that this is lactose, and it's got galactose and glucose. So basically, it will attach to lactase, so you can say that this is lactase. So the enzyme could be is lactase, And basically, when lactose bonds to lactase, you get the complex, the enzyme substrate complex. And basically what will happen here is the enzyme will help the chemical reaction to break lactose into galactose and glucose, which would be what you might see here. This could be lactose this would be lactose and this would be galactose and glucose. So you'll see when you're looking at anything talking about enzymes, something known as induced fit. And basically, induced fit is going to be when that substrate binds to the active site, you'll see that the enzyme fits very, very closely to that substrate. It binds very snugly around it. And induced fit is one of the many reasons why enzymes only work with one substance, because other substances aren't going to be able to create this induced fit. Now, as you've learned in your lab, enzymes' activity can be affected by temperature, pH, and chemicals. So if you remember back to the activity where you guys formed a huge human protein, think about this. An enzyme is a protein. So an enzyme is going to have R groups, just like a protein, because it is a protein. And those R groups will absolutely be affected by the environment. So if you have an R group, for example, that is highly basic, what do you think might happen if it's put in a very acidic environment? Well, those R groups are going to be highly attracted to that, and it's going to cause it to denature or come apart. Likewise, if we increase the heat, it is going to denature and fall apart. If it is too cold, it's also not going to function well because you're going to lose some of the kinetic energy because you're going to be losing some of the heat. Likewise, chemicals can greatly influence the function of your enzyme. So for example, if you have an enzyme that is formed because of the interactions between sulfur groups in the R groups, those disulfide bridges could be absolutely destroyed by any chemical creating sulfur. Ions can absolutely affect this as well. So some groups actually affected, tested to see, well, what would happen if we put salt in? And they saw something very interesting. It can help increase and then decrease, because at first those ions can actually help the enzyme, and then they can decrease it. Something else that you should absolutely know is about the presence of cofactors. So cofactors are basically non-protein enzyme helpers. So these are not proteins, but they're enzyme helpers. These can be minerals like zinc, iron, and these things absolutely can help the enzyme to function. There are also coenzymes, and we'll really be getting into coenzymes when we get into cellular energy. But these are basically organic things, whereas zinc and iron, not so much. So these might be things like vitamins. So let's look at how some of these things can work, and let's look at enzyme inhibitors. So there are two basic types of enzyme inhibitors. Competitive inhibitors basically would be a substance that can mimic the substrate, and it can bind to the active site of the enzyme, and then when the enzyme tries to bind, it can't fit because there's something else there. Then there are non-competitive inhibitors, and what they'll do is they'll bind to another part of the enzyme, and it will change the shape of the enzyme. And this will change the active site so that the active site is no longer functional. So let's look at a picture of this. So this is basically a normal enzyme, and here's our substrate that can fit in here. You can get a competitive inhibitor which can mimic mimic the substrate and bind in its place, so then the substrate, it can't go in, it can't be broken down. Then we can also get a non-competitive inhibitor. This means it's not competing directly with the substrate, but when it binds, it drastically changes the shape, or in some cases it's not even that drastic, but it's enough where the substrate can no longer bind. In both cases, the enzyme no longer functions on the substrate. Okay, let's talk a little bit more about regulation of enzyme activity. So enzyme inhibitors, like those we were just talking about, can actually work very well to regulate the function of the enzyme. Now, sometimes you're going to find inhibitors in drugs, medicines, and things like that, and some of them can be quite bad. However, they also work to help your body to function the way that it's supposed to. So basically, it can allow us to switch certain genes on and off, to allow us to have a certain enzyme present or not have it. And specifically, allosteric regulation allows us to turn an enzyme on or off. So basically, if you put a non-competitive inhibitor in place, it will make the enzyme not function. So let's take a look here. Oh, and you should know some vocabulary. Activator. The activator is going to be something that makes the active site stable and allows the substrate to bind to it. The inhibitor will stabilize the inactive form. So inhibitors, again, could be competitive or non-competitive inhibitors. And then you also sometimes will see cooperativity, where one substrate will trigger a shape change in other active sites. And this can actually increase the catalytic activity of the enzyme. So let's take a look at this diagram. I think this will make more sense. Allosteric activators and inhibitors. If this is the active site here, so this would be an active site, and you'll see that there are 4 on this particular molecule, and then there are regulatory sites. So basically, the activator can come in, and so it's set in its active form. It's going to fit the substrate. Now, you can remove this, and then when this is removed, then there's the inactive form, and that's not going to function. The inhibitor can come in and make it not function as well. So this is a stabilized inactive form. So this is stabilized active form, stabilized inactive form. And this is cooperativity, which basically you have your inactive form. The substrate will come in. That substrate will bind, and now it's stabilized in an active form and can bind to other molecules. So, and this is really an example of feedback. And there are 2 types of feedback that you need to know about, and their names are kind of misleading. There's positive feedback and negative feedback. But positive doesn't necessarily mean good, and negative doesn't necessarily mean bad. So positive feedback loops, these are really runaway cycles. Basically, in a positive feedback loop, the input will cause an output, and that output will basically cause an even greater input. So in this case, an input will create an output, okay, and then that output creates, becomes an input, so that the output is an input. I know this is confusing. That increases further output. So this example of melting of permafrost, so it will release methane, which will accelerate global warming. So the input in this case would be, the input is increased heat. The output is melting of permafrost. But then the melting of permafrost also has the release of the methane, which causes an increase in heat. And then that increase in heat will cause more melting of the permafrost, which will release more methane, which will increase the heat, which will increase the melting of the permafrost, which will increase more methane, etc., etc. It is not self-correcting. It can go out of control. So it just causes increase upon increase upon increase, if that makes sense. Hopefully my verbal explanation made more sense than what I typed. Negative feedback loops occur when basically the input can regulate the output. So a good example of that is an example of moose and wolves. So let's say in the system you have a lot of wolves, and wolves feed on the moose. Well, the input would be the wolves eating the moose, and then you'll get an increase in wolves. When you get an increase in wolves, you get a decrease in moose. Then, because there's a decrease in moose, not enough wolves will get food, and so then some of the wolves will die, allowing the moose population to increase. And as the moose in population increases, you get more wolves feeding, and it totally self-regulates. Totally self-regulates. So feedback inhibition. Basically, feedback inhibition will be when you get the end product of a metabolic pathway and it shuts down the pathway. So this would prevent the wasting of chemical resources. So for example, let's say that you have an enzyme that breaks down, I don't know, let's say lactose. So let's say you have lactase, and let's say that lactase turns on in the presence of lactose. So you have lactose, the lactase breaks it down, and then the products, do you remember what the products are? Do you remember what those were of lactose? You get galactose and glucose. So let's say the galactose and the glucose bound to the lactase, shutting it down when there's no more lactose in the system. What kind of feedback would that be? And then as soon as you get more lactose, those things are released, they're used. Would that be positive or negative? It would in fact be negative. Okay, so let's take a look at this. So here's the active site of enzyme 1 is no longer able to catalyze the conversion of threonine to its pathway. And don't worry about all these details, but understand really how it works. So, okay, in this case, we've got isoleucine. Okay, and if you don't remember what isoleucine is, look it up. Have you looked it up? It's an amino acid. So this will bind to the active, so this binds to the allosteric site, and when it's bound, the substrate cannot bind. When it's released, the active site is available. So that's an amino acid. Threonine is also an amino acid. So this is the substrate. So when the substrate comes in, it binds, then you get our intermediates, and then you get the product, which is isoleucine. So isoleucine can bind to this when there is no threonine present. Once, and it will bind until it needs to be used by the cell. When it's used by the cell, then we need more of it. So then the enzyme will allow threonine to come in. The threonine comes in. We make more isoleucine. Once we have a bunch of excess isoleucine, it will be bound to this site, stopping the production of more isoleucine until it's needed again. Hopefully that makes sense. So here are some checks for understanding. You should be able to know what the first and second laws of thermodynamics are. And obviously with all of these, you want to be able to relate them back to biology and give examples. What is catabolism and anabolism? And what are specific examples? If you're breaking down glucose in cell respiration, is that exergonic or endergonic, and how do you know? You should also be able to label an enzyme, its active site, its substrate. And I would honestly add some other things into that. I would add in allosteric regulation. I would put in competitive inhibition, non-competitive inhibition, and discuss how those things can affect the enzyme. You want to absolutely describe what induced fit is and how that works, and what types of factors can affect the function. The other question that's not on here that needs to be is you should understand the differences between negative and positive feedback with examples. Now, I didn't give you many examples because I really want you to look in the book, or you can even look online for some specific examples, because I think trying to determine which example fits which type is going to help you to further differentiate between these two types of feedback. One common mistake on the AP exam is mixing up the two types of feedback. I will give you a hint. You can look at women and hormones in pregnancy and childbirth to help you with positive feedback, because there aren't a lot of examples in positive feedback in humans, for example. I'm sorry if parts of this were confusing. This is the very first video that I've made, video lecture, so it was a little nerve-wracking for me. Do not hesitate to email me if you have any questions at all. And I look forward to seeing you guys in class.